It's important as a student and future professionist to know all the equipment and materials that are useful in the laboratory, and more important, how to use everything in order to make good practices. This video explains how to use properly the materials that are in the laboratory, especially how to use micropipettes. In the activity 1 and 2, we draw in a table every material and equipment that we can find in laboratory in order to be more acquainted to it.
Each team member was practiced pipetting technique. Measurements were performed using 3 pipettes of different sizes. We took a volume of deionized water, indicated in the binnacle, and then waited to observe the weight was taken. Finally, the standard deviation of the data is determined to notice the error.
To continue practicing pipetting technique of each member, an activity was performed in which we added an amount of volume in triplicate in a microtube performance, subsequently recovering the total volume and the recorded performance to verify that the measurements were correct. At the end, the average and standard deviation of the measurements is obtained.
For each sample taken, we registered the weight in order to know if we were taking the correct amount of water. For the same activity, we calculated the average and standard deviation in order to quantify the error. In the second activity, we calculated the average and standard deviation. As we can see, there was no error in the first triplicate, but in the other two samples we can see a slight error. In conclusion, this practice helps each member to know the materials and equipment that are in laboratory and also how to use properly the micropipettes.